Hi, and welcome to this presentation titled Accelerating Workflows for Calprotectin and Complement Biomarker Assays with Nanoliter-scale Microfluidic Immunoassays. I will be presenting to you a few tests that we have developed for Calprotectin and Complement C4d and TCC for nanoliter-scale immunoassays. My name is Ali, and I have quite recently taken up the role as Product Manager for Complement and Calprotectin portfolio at Svar. I will be presenting the topic for you today, and if you have any questions, please feel free to type in the chat as we go along. Before I continue, I would like to say a few words about our company. So, delivering answers in life science is basically what our company does. We provide you with assays and services required to get the answers that you need in order for your projects to succeed. And Svar is a Swedish company with a long history within life science field as supplier of kits, solutions, and services. The company quite recently, a few years ago actually, adopted the name Svar. Some of you might even remember our old name, Eurodiagnostica. Our aim is to be your go-to partner to get reliable assay solutions and services to get to complete your projects, both in clinical diagnostics and in research and drug development. This is actually reflected in our name, as Svar literally means answers in the Scandinavian languages. What our company provides can be easily split into two bulks. The first one being the products that we have, which are our CCP assays for rheumatoid arthritis, our cell-based reporter gene cell lines called ELISA, and our complement portfolio, and finally our Calprotectin solutions. The second bulk would be the services that we can provide through our bioanalytical and diagnostic services, as well as the services provided to customize the ELISA assays with the specific targets that you need for your projects. This goes from everything, this involves everything from planning and research to the actual development and even storing the ready-to-use assay cells at our facilities. So just to give you a visual presentation, for each of the steps in drug development and clinical application of these drugs later on, Svar has tools and technology that can be applied. And where the actual tool or the assay is missing, we can use our knowledge and expertise in the field to develop new assays, new markers, with new markers that will fit your need. So the Svar products and services can be split into four main groups. The ELISA cell-based solution is our off-the-shelf and custom-made assay-ready reporter cell lines for functional assessment. They can be used for drug candidate screening, potency assays, neutralizing antibody assays, and much more. It's a great technology allowing you to carry out your experiments with and achieving reliable results and with high reproducibility. Our complement portfolio have several assays based on ELISA technology, and they are either quantitative or having a functional assessment aspect to it. When it comes to our bioanalytical services, they develop and perform pharmacokinetics, toxicokinetics, pharmacodynamic, and immunogenicity experiments for our partners, as well as support through consultancy services. Bioanalytical services are currently working under the GLP standard, but soon they will be upgraded to GMP, and they can be an even more well-rounded partner. And finally, Calpro, which actually is a Norwegian biotech company Svar acquired a couple of years ago, have a focus on a single biomarker, Calprotectin. This biomarker is used quite broadly in clinical studies and also in diagnostics and distant management of patients with inflammatory bowel diseases, such as ulcerative colitis and Crohn's. For this presentation, our focus will be on complement and the calprotectin. But first, why nanoliter-scale solutions? So in drug discovery and drug development, especially in the early stages, there is a lot of pressure to identify the correct compound. Nanoliter-scale immunoassay technology is made especially for that. It improves the process and allows for quick identification without compromising the quality of the experiments. Nanoliter scales allows for high sensitivity and precision by reducing the handling required. Number of tests performed and the assay time itself reduces the assay time comparable to other standard methods. One of the main benefits with nanoliter scale immunoassays are without a doubt the ability to reduce sample and reagent volume used. After all, these runs require nanoliter scales. This means that even with limited sample or limited reagents, you can run a full set of runs. The tests we will look at today are a bit do-it-yourself type of tests, in the sense that not only they can be assembled to a kit according to your needs, so you can assemble a kit customized for yourself with only the reagents that you need for your experiments and the compounds that you need, but you can also adjust the suggested protocol according to your need and optimize it much easier than say ELISA type of assays. Lastly, I would like to mention that each instrument provider will have specific technology that will give you additional output that will be of great benefit. So nanoliter scales make screening and analysis of large number of samples rather effective and easy, both from time and cost perspective. So a little bit about the complement system. The complement system was discovered in the late 19th century, and for the past decade or so, it has been a really focus point with a lot of focus on how to use this as drug target to treat different diseases. So complement is a part of the innate immune response and consists of a cascade reaction that results in one of the three following actions that occurs in order to get rid of invading pathogens and debris. And yeah, the very first one is the anaphylatoxin properties. So some of the breakaway fragments from these main molecules, they are very effective in attracting and activating cells such as neutrophils, monocytes, macrophages, and attract them to the site of inflammation or infection. They also assist in increasing the vascular permeability, hence making it easier for these phagocytic cells to reach the area of infection and inflammation. The second thing they do is that they kind of light up the target cells. They will tag the intruding cells or the bacteria or the debris floating around, so that they can more easily be identified and removed by the phagocytic cells. And lastly, the complement cascade system, complement cascade ends up in forming a membrane attack complex, basically forming pores on cell surfaces of invading cells, causing the cells to burst due to high influx of water. Now, although presented quite simplified in this diagram, it is a bit more complex and there are a lot of more molecules involved. It consists of more than 30 proteins, and the assembly, in order to achieve anything in this one, needs to be in a specific order within a specific time frame. For some of the activated molecules here have a half-life of microseconds, and the reactions needs to happen fast. There are three main pathways to activating the complement system. They are termed the classical pathway, the lectin pathway, and the alternative pathway. The targets for the assays that we will look at today will come from the classical and lectin pathway, which is the C4d encircled at the top left, and the terminal complement complex TCC all the way at the bottom. Just to point out the fact that complement is a really focused area for drug development nowadays. This diagram, published in 2018, shows just that. There are a lot of different drugs that were tested in 2018 and more today, targeting various aspects of the complement system. And this is increasing in line with new indications that appear. As research is progressing, more and more diseases are identified as being caused by malfunction of the complement system, or that can be actually treated by doing something with the complement system. So the complement C4d. Now the complement C4d molecule is an activity marker for the classical and lectin pathway. Now C4d is a breakaway product of C4, which is located at the very top. C4 is actually cleaved either by C1 complex of the classical pathway or the MASP1, MASP2 complex of the lectin pathway. So if C4d is detected in a sample, this will indicate that the classical pathway or lectin pathway was somehow activated. Now, C4 is normally cleaved into C4a and C4b, and C4b will stick to the cell surface and bind C2. Now, if this bonding to C2 fails within a really short time frame, the complement factor I will cleave the C4b and make it inactive. This new molecule is termed I-C4b. Now, factor I will continue to chew on this molecule and cleave off a tiny bit. So the I-C4b will be cleaved into C4c, which is a soluble molecule, and a C4d component, which will remain on cell surface. Now, seeing that this is an endpoint of a cleavage sequence, it makes a good biomarker and also a target to detect if any drugs have affected the classical pathway or the lectin pathway. Now, the actual biological function of C4d is not known, but it is actually used as biomarker for various conditions, such as antibody-mediated graft rejection, and also used as a tool to predict and monitor flares in SLE, systemic sclerosis, and ANCA-related vasculitis. And also, there are some research being done where it shows potential to be a lung cancer marker, as well as a marker to show response to chemotherapeutic treatment. Now, although C4d is not yet found to be associated with any diseases specifically, its precursor is. Now, C4 deficiencies are often related to recurrent and increased risk of infections, and also various systemic autoimmune diseases such as lupus and rheumatic diseases. So Svar already have ELISA for this fragment of the complement system, and these antibodies were then used to create an assay for nanoliter-scale analyzers. And as our collaboration was with Gyros, the initial testing of basic application was done on their instrument, which I will present to you next. The assay range for the complement C4d Flexi is, as you can see, up to 500 nanogram per milliliter. Any concentrations above that obviously need to be diluted into the correct range, as this is the linear area. When looking at the CV and reproducibility of it, we see that the values are quite good for 40 microliters and up. 40 and below, or actually 30 and below, when pushing down on the lower limit of quantification, we see that the CV is somewhat higher. But for the rest, it is rather low and quite good. In order to test for recovery, serial dilutions were made of different target concentrations. Now, the theoretical targets for the dilutions were based upon the least diluted sample, which is the blue one, which is 25-fold dilution. The others were 57 and 128 respectively. And here you can see that the recovery is quite good and quite acceptable results. Another thing which is important, especially for the complement C4d molecule, since it is a precursor, it has a precursor in C4b and C4, is that one is assured that there is no cross-reactivity to its precursors. So we did some experiments to show that there is no cross-reactivity, as you can see on the bars, to C4 or C4b. C3 was also tested because that is really abundantly present in circulation. And also C5 and C9 were tested for cross-reactivity to the antibodies, and it showed none. So the antibodies are quite good, and they bind to the neoepitope site of the C4d, which is revealed once the C4c is cleaved off. So all in all, quite a good assay to use on the nanoliter-scale solutions. Moving on to the complement TCC. So the complement TCC is one of the two endpoints of the complement cascade reaction. All three pathways connect at C3 point in the cascade, and once C5 is cleaved to C5a and C5b, the remaining components of the terminal complex can be formed. Now, if C5b is bound to a cell surface, the addition of C6, 7, 8, and 9 will form a membrane attack complex. And result in a pore created in the cell wall it is bound to. If C5b is not attached to a cell surface, the complex will bind a molecule called S protein to form a stable structure, and it will bind the remaining complement molecules 5, 6, 7, and 8, and 9. And this will then be in solution. That's why the TCC is sometimes also called the STCC, for soluble terminal complement complex. Identification of the TCC shows that the complement system somehow have been activated. Now, it doesn't say anything about which pathway was used to activate this, and hence the initial source of activation, but it does say that the pathway was activated. Now you can combine this with C4d to narrow down the activation to either classical or lectin pathway, or the alternative pathway if the C4d test shows no activation. The actual function of TCC is not clear, or the soluble TCC is not clear, but it is used as a biomarker for acute injuries such as trauma, sepsis, and myocardial infarction, and in chronic inflammatory diseases as well. It is also used as a predictor of adverse reactions from medical devices. The complement TCC Flexi was also tested in the same manner as C4d. So the first thing that was done was to identify the upper limit of quantification, which was 400 nanogram per mil for this assay. And here you can see that the CV is quite low all across, pretty much, apart from one outlier at sample 7. Other than that, it's quite good CV, both within runs and between the runs. The same goes for the recovery. As with C4d, the lowest diluted sample, so the tenfold dilution, was used to calculate theoretical standard for the other dilutions, and most of the samples, most of the dilutions showed really good recovery, apart from the 80-fold dilutions that was somewhat higher, but the remaining runs were quite good. So moving on to calprotectin. The question we will first look at is, how is calprotectin connected to the other two tests? Now, as mentioned in one of the previous slides, one mode of action of the complement system is to act is to attract phagocytic cells. This is done by the anaphylatoxin C5a, which will recruit cells to the site of inflammation and infection, as well as increasing the permeability, vascular permeability. Now, C5a will especially trigger eosinophils and neutrophils. And for neutrophils, their cytosol is made up of 60% of calprotectin. Now, upon turnover, that calprotectin will be released into the surroundings, and then it can be measured, and increased levels of calprotectin will hence reflect upon increased level of neutrophil activation at any given site. So calprotectin is a part of the innate immune response, and that's why it is such a good biomarker for inflammation. It is a general marker, similar to CRP, C-reactive protein, with the added benefit that it is not synthesized in any way, just released. Calprotectin is actually really stable, so this makes it even more of an ideal marker. To give you some idea of its robustness, if a patient would have inflammation or infection in the throat where calprotectin is released and swallowed, it will actually go through the entire digestive system and come out in the stool, and it can still be measured in the stool. And it can even stay in a stool sample for up to 5 days, which is quite amazing for a molecule to go through that kind of treatment. Currently, the main clinical application for calprotectin is seen within the gastro field and as a biomarker for IBD. But there are a lot of publications done using this biomarker in plasma and serum from the mid-1980s, when the biomarker initially was discovered. A fun fact is that those who initially discovered and characterized and even named this molecule later founded the company Calpro that is now part of Svar Life Sciences. Anyways, apart from the clinical applications, it is widely used in clinical trials for IBD studies. And one of the reasons is that it is shown to have really good correlation to predicting mucosal healing with in patients with inflammatory bowel disease. So by using calprotectin, they can avoid using other invasive examinations such as endoscopies and colonoscopies. With the recent developments in technologies and stuff, these testings for stool fecal calprotectin can actually be done by the patients at home as well, making it even easier to collect the data for studies. Now, as mentioned, there are a lot of studies done on calprotectin apart from IBD, and the main field of focus have been rheumatoid arthritis from all the way from the 1980s. But calprotectin is present in other bodily fluids, and it can serve as a marker for diseases in those systems as well, such as present in synovial fluid, urine, and saliva. The best use of this marker is obviously, as any general marker, is that it is tested in closed systems. So if you would test urine, you would know something is and would have elevated calprotectin levels. There would most likely be something with the urinary tract or the bladder or maybe the kidneys even. And with saliva, it would be something to do with the mouth, the tongue, the teeth, and so on. So by narrowing down and using liquids from closed systems, calprotectin can be a really good and strong biomarker for inflammation. The calprotectin flexicate was tested the same way as the preceding two, and however, this marker, as it also is used in stool samples, was also tested using blood and stool samples for the flexi testing. The range for the calprotectin is higher than for the C4d and TCC. It actually is up to 1500 nanogram per ml. And from the CV, and you can actually see that it is quite low. So it's really a good test to use. The only outlier here is the first sample, which is really pushing the boundary on the lower limit of quantification with a mean concentration of 3 nanogram per ml, when the lowest limit is 2 milligram per 2 nanograms per ml. Apart from that, the other values for CV is below 5.4 for within the runs and less than 6.2 for the between the runs. A recovery run was done here as well, having different levels of target values and different dilutions, as the target values differ so much. And you can see that all across for the 3 that is shown here, they were quite good. The recovery was quite good and within acceptable ranges. So this actually brings me to the end of my presentation. I would like to just wrap up with sharing with you that if you are interested in learning more about our Flexi kits or our iLight reporter cell lines or complement portfolio or any of other Svar assays and lab services, please do visit our website at svarlifescience.com and connect with us. And we can take it from there and see how we can help you. Other than that, thank you for your attention and your time, and have a very nice day. Thank you.